recorded. And so this can be viewed at a later date. So if you need to step away during the middle of the event, don't worry, this will be archived for recording. And so for today's discussion, we would like to talk about the topic of pathogen surveillance. Right. So COVID-19 is something that we all are very familiar with, and probably something that's very personal to all our lives, especially during the height of pandemic during 2020 to 2022. But as we approach the halfway point of 2024, this seems like something that's from the very distant past and has forgotten from most people's mind. While our lives has been by and large returned to the post-pandemic norm, one of the key questions that's still lingering on our mind is how do we prepare for such an event in the future? So what happened during 2020 to 2022 doesn't repeat itself again. So really, pathogen surveillance is the leading frontier to answer such question, right? So pathogen surveillance can help us answer a question like, how do we help us investigate spread and transmissibility of a threat? It helps identify variants of concern, such as greater transmissibility, immune escape. It can tell us answers like, what's the variant, when and where does it circulate, why is it a risk, and who in the population is the most vulnerable. It can also help us design and optimize medical countermeasures, diagnostic tools, vaccines, and corresponding countermeasures, and take appropriate actions toward the threat. If we have these understandings during the early stage of the pandemic, it will certainly help reduce the impact of the threat, both in terms of public health, as well as reduce social and economic impact. And for today, we are really excited to assemble this panel of experts to share their experience in the various biosecurity program. And before we start, I'd just like to give a quick introduction about myself. So I'm one of the moderators today. My name is Alex. I'm a senior scientist at Ginkgo Bioworks, and I primarily works on microbial engineering and specialize in synthetic biology and production of natural product. And we'll have another kind of co-host today. Qixin, why don't you give us a brief introduction?
Sure. Hi, good evening, everyone. Also, good morning to audience from Taiwan. My name is Qi, and I'm now a postdoc in chemistry at MIT. I also have the privilege of serving as one of the moderators today. And a little bit about me is that I am also a member of the Superfound research program, which I work on detecting the human carcinogens in the drinking water. So, and I believe that the topic we're discussing today are crucial real-world problems that in need of solutions. So without further delay, let's get started with introducing our two distinguished speakers. Alex?
Yeah, so for today's panel, first of all, we have very excited to have Robert Morfino joining us today. Robert is the Senior Director at Ginkgo Bioworks as part of now the rebranded Ginkgo Biosecurity. Robert comes in with a wealth of experience in program management, spending close to 7 years at McKinsey and also spent more than 4 years now at Ginkgo Bioworks. And he has a wealth of experience both in the technical sector as well as project management, and is also a very good storyteller based on a brief conversation I had with him earlier. And we have the second panelist, Dr. Shu-Yu Hsu, and she is a scientist at Verve Therapeutic. But Shu-Yu is here today to share with us her works during her PhD in University of Missouri under the supervision of Dr. Lin Zhonghe. And her work there is primarily based on wastewater surveillance of COVID-19 viruses. And so I think the combination of these two panelists, both from the project management perspective, as well as from the on the field, in the lab academic perspective, will make a very dynamic and exciting conversation. So before we start, I'd just like to have a quick housekeeping business. So for today's agenda, we'll have both Robert and Shu-Yu share a brief presentation on their work. And after that, we'll have an open panel discussion to dig deeper into some of the more interesting aspects of biosecurity and pathogen surveillance. So please reserve all your questions till the end, where we will have a Q&A session for the audience here. You can either leave your question in the chat, and me and Chi will help monitoring the chat room to help funnel your question through, or you can just raise your hand and turn on camera and ask the question yourself too. But please just remember to save all the questions to the very end. And so before we start today's event, I would just like to do a group photo. So if you don't mind, you can turn on your video camera, and we will do a photo shoot before we get going here. And we will do the group photo in 3, 2, 1. Perfect. Thank you so much. And with that, I will pass the baton to Robert, and take it away, Robert.
Fantastic, Alex. What an introduction. I really like the distinguished guest that I think Chi called me, so I'm very honored. But most importantly, I'm honored that you guys are choosing to spend your Friday night or Saturday morning with us and, listening to this. That means that the expectation in the bar now is very high. So I'm very nervous about that, but I'll do my best to make it worth it. So I will share my screen because I have a few pages. Alrighty, so building a global bioradar for early pathogen detection and characterization. The first page is a standard page that we have at Ginkgo describing a little bit our business. So there's essentially 3 components in our business, in our company. The first is the foundry, which is essentially a highly automated, high throughput lab, if you will, where we essentially perform most of our wet lab activity. The second piece is our code base, which is in many ways our dry lab. So it's essentially a large database that contains all of the data from all of our experiments and work that we've done so far. And we use that database as the basis for essentially in silico design of our products. Obviously, Alex here knows more than I do about this. So if you have questions on these 2 first section, Alex is the right person to ask. I'm focused on the biosecurity business, which is essentially, we'll talk about this more, but it's an integrated end-to-end biosecurity platform. And we'll deep dive into this in a minute. So I'm not going to spend too much time on this, but just this is at a high level what we do at Ginkgo. Our platform, our business, our company is really a platform technology. And so we can apply to multiple verticals. That's why we serve multiple clients, partners. We also have what we call opcos, that are essentially company that leverage our technology in many fields. You can see here, right? Food, agriculture, pharma, materials. So essentially, really the idea here is that biology can be a new way of producing anything we need. I'm sometimes a little bit, ambitiously, we like to talk of it as a new industrial revolution, and we really believe that this new way of production is going to be much more sustainable, much more efficient, and really what is needed in the future of our society. Now, in the biosecurity division that we just discussed, there's 3 big capabilities that we are working on. The first is essentially what we call the monitor, so the detection platform. It's essentially a collection of capabilities, expertise, and tools that help to launch detection programs around the world. The second piece is around characterization analysis, where we use sequencing and bioinformatics, as well as epidemiological modeling. And finally, the third piece is the response, where we support our client to think about what kind of response mechanism they should use in a certain situation. So we've been working over the last 4 years to build capabilities across these 3 big pillars, if you will. Now, if we deep dive into the first one and the second one, we actually call the first one BioRadar. And as you can see, really the key here is to have a platform that is multimodal. So we're not wedded to only one sample type or only one source of data. We try to really be modality agnostic. We handle nasal samples, wastewater samples, air samples, surface samples, and so on. We really believe that the power of the platform is leveraged when you can deploy it in multiple sites. And so these are some of the locations that our platform has been employed in. So airports, mass gathering, communities and congregate settings, hospital and nursing homes, agricultural sites and neighborhoods. We really believe that the strength of this platform is when we get a diversity of sites. And finally, and most importantly, we cannot only focus on COVID-19, we have to be multi-pathogen. In fact, we have to be pathogen agnostic. And so we're building the capabilities so that our samples can be used to detect any pathogen. And that's actually what our clients also want. So this is our bioradar. So essentially, it's our physical, if you will, infrastructure. Then we have our data analysis platform. We call it BioInt. Actually, we just rebranded it. Now it's called Horizon. But essentially what it is, it's a platform that analyzes hundreds of data feeds. Some of the data feed are our own data feed that we have produced through our bioradar. Some other data feed are open source data feed, but what we do is that we structure those data feed and we mine them using AI, and we're able to then categorize that information and provide our clients with early warnings and information about the threats and the outbreaks that we're seeing, but also we're able to model and forecast potential outbreaks. So those are the two big pieces, if you will. There's the physical aspect of our platform, and then you have the digital side of our platform. Now, if we focus on the physical, we essentially, ultimately, we're trying to build a network, and each of the node in the network has to be equipped with enough capability to produce the relevant data. And what we do to produce this data is that we deploy this end-to-end workflow that starts with the collection of samples, goes through the processing and enrichment of the samples, the extraction of the RNA and DNA, the sequencing preparation, the sequencing itself, and then the analysis and the alert and the modeling. So essentially, anywhere we go, and you'll see in a minute a map, each of these nodes is equipped with this. It doesn't mean that all the capabilities have to be locally, right? Some capabilities have to be there, like you need a sequencer, but the bioinformatics doesn't have to be locally at every single node. It will be very inefficient. In fact, we try as much as possible to use cloud-based solutions and centralized SMEs. And that's because ultimately the system has to be distributed, but the capabilities don't have to be replicated everywhere across the network. On the BioInt, you can see here a couple of screenshots of what the platform looks like. Essentially, it's a dynamic solution that enables you to get all of this data feed in one place. And this is very important because today there's a lot of surveillance programs across the world, but a lot of this data is very scattered, and there's no single place where you can actually analyze it. So that's what we're trying to solve for. We're trying to make this platform really integrate all of this data feed. And by the way, we're trying to open the platform to as many people as possible so that they can contribute to it. A little bit of the story, the history of how we came to be. So back, if we go back in 2020, in February, I was actually interviewing with Ginkgo. I really wanted a job with Ginkgo. I had left McKinsey, but I wanted to join this exciting startup, and everybody was talking about it. So I interviewed with Ginkgo in February, and then in March something happened, right? This news about the virus happened, and I finally got the job. But very soon the company, started to shut down, like every other company, except for one thing that continued to happen, and it was an effort around how can we support the response to this unknown virus. And obviously our founders, they wanted to do it mostly because they wanted to help. They really believed that the company had a role to play here, given that we are a big player in biotechnology. And so initially what we did is that we leverage our lab capacity, both the PCR and sequencing, to offer large-scale testing to schools in Boston and elsewhere in the country, but also employers, prisons, any facility, honestly, that needed to stay open because they were essentially performing some critical work. So that's how we started. We started essentially providing testing services to thousands and thousands of schools, and it wasn't really meant to be a long-term business. It was just supposed to be a way for us to help. Now, very quickly, we realized that actually this idea of providing large-scale monitoring was actually a need that could exist even beyond the pandemic. And in any case, was a need that, you know, no one really was providing in a systematic and professional way. And so in 2021, we launched our first non-testing, if you will, product, which was a program focused on airports. We started with CDC. We started a small pilot in 2 airports, Newark and JFK. Initially, we started swabbing passengers coming from a couple of countries where we had seen new variants emerging. So we wanted to kind of provide CDC with fresh samples that we could sequence and characterize. And then very quickly with Omicron, there was immediately a need to expand the program to cover many, many more countries. If you remember, at the time, one of the first warning came from South Africa. So we started monitoring flights from the southern region of Africa, but then very quickly Omicron was everywhere. And so we very quickly developed this network of airports in America. And still today, we are testing passengers, but also now we do wastewater from planes and from airports. So that was our first, let's say, big program with the federal government. Before we were mostly working with either schools or state authority, public health authority. In 2022, we expanded into environmental testing. And also started publishing with CDC. In 2023, we expanded to multi-pathogen. Obviously, the focus until then had been SARS-CoV-2, but very quickly the natural expansion was to move to beyond that. And then we expanded to agricultural settings. We also launched our BioWin platform. And in 2024, we actually went even further, right? We expanded into something that is not really obvious for us, and it's high-risk substances. And the reason is ultimately, if we're trying to assess and help public health to assess the community, the health of a community, then why wouldn't we also look at the other side of the health of a community, which is the chemical biomarkers, and in this case, biomarkers of illicit substance consumption. So really, you can see how this unit has evolved from something that was supposed to be a temporary response to something that is here to stay and is not pandemic dependent anymore. Here's a map of the network today, countries where we have active programs. Some have not been announced, so they're not shown in this map. This is just what has been announced. We also launched a genomic center similar to what we have in Boston. We launched one in Qatar, in Doha, recently, so that we are able now to process samples in multiple places. Although in reality, our philosophy is that we have to use local capacity. So remember, we just talked about the nodes and the end-to-end. We much prefer to use local labs as much as possible, but sometimes when there is no local capacity or capability, at least in the short term, we use the lab in Qatar or the one in Boston. We already talked about how it is really important if you want to have a holistic surveillance system to cover as many different locations as possible, and especially those where you're more likely to detect something new, right? Typically the interfaces between countries, so the borders, but also interfaces between the natural world and the human world, right? So farms, for instance, or wild areas where humans may interact with animals and so on. Now we're going to have a few case studies of programs that we're running. The first one is our CDC TGS, traveler-based pathogen genomic surveillance program, where essentially we are, as we said, active at 10 international airports in the US, and we are collecting samples from passengers and planes. This is important because this is very important because it provides important information to the CDC, our public health agency authority in the US. For instance, this program was the first one to detect BA286 from Asia, Japan in particular. We were one of the first to detect JN1. We were the second in the US. We were the first one to detect BA2 and BA3 when Omicron first started. And then we are actively monitoring for Mpox, especially Clade 1, which is something that has caused a lot of concern in the public health circles. So you can see it's a program that has been consistently providing early detection and characterization. And that's the reason is very simple, right? It's because instead of waiting for the pathogen to show up in a clinic within a community, we catch it as it enters the country. And so by definition, we're going to be early. And this really is the idea of this kind of program. The other thing is that it fills gaps in global surveillance. So not every country is performing surveillance in the same way. Not every country reports data in the same way, right? There are some really good actors, but there are also countries that don't want to share. That creates essentially black spots in the global map of surveillance. And this program, by being essentially active in airports, provides coverage and insights into those spots that don't report. And so that's really a cool thing about the program. And by the way, when we talk about SARS-CoV-2, we may talk about dark spots. When we talk about other pathogen, we talk about a dark map of the world, because very little surveillance is done for other pathogens, right? If we think of one that has been in the news recently, RSV, there's incredibly little surveillance for that. And so really this kind of effort is very helpful. This is one example that really makes it tangible. How do we capture those samples? This is an example of a device that we have designed to collect wastewater samples from planes. A reason that we're doing this is that we look at planes as a proxy for passengers. And the other thing about planes is that it kind of gives an indication of where the sample is coming from. And so that essentially enables us to get a sense of what is coming from where. So this is one of our colleagues, by the way, one of our Ginkgo colleagues here next to the ground handler, who is the person that does the servicing of the plane, explaining how to use the devices. This was one of the first collection that we did 2 years ago, but since then we've done thousands of those collections. So if you take a plane and you come into the US, please know that sometimes your plane may be sampled, the wastewater from the plane may be sampled for further analysis, but you don't have to worry. We only look at pathogen. We don't look at anything else. One example that I was saying, so all of the data that the CDC gets from this program gets analyzed and reported. So if you go on the CDC website, you will find the data in real time that we are producing, published, because obviously the CDC really believes in the importance of sharing this data with the public. And up until today, we've had more than 500,000 participants from 135 countries. So it's really one of a kind program, I would say. Other example, farms. So that's another big topic, biosecurity in the ag world. In this specific case, we were looking at antimicrobial resistance, but more in general, we really believe that there's a reason for doing pathogen surveillance in farms. And obviously the recent events with highly pathogenic avian influenza demonstrate that this need is more present than ever. And in fact, I think that right now people are realizing that the fact that we have minimal surveillance among farms is a big gap in our public health system, in our pathogen surveillance system. So this is changing slowly. But in this case, what we did is that we were focusing on a farm in Australia, looking for markers of antimicrobial resistance. We actually found a lot of them. But I think what we really demonstrated was the feasibility of doing this at scale. We really believe that in the long term, this will be done everywhere and at scale. Some example of how we collected those samples in a farm. I thought that those pictures were pretty fun to put because, again, it makes it very tangible, right? What does it mean to do biosecurity? Yes, there's a fancy part of analyzing data on a computer, but there's also the less fancy part of going to collect those samples in some pretty tricky places. You can see here some examples of what happened in Australia. All right, hospitals. So hospital is one that is really close to my heart, because when we think of the next best thing in pathogen surveillance, we think of hospitals, right? Because ultimately they are this point of aggregation for a community. And if something is circulating in the community, most likely it will get into the hospital. So the hospital can be a really important node in this global pathogen network, pathogen monitoring network. But also, we believe that there's ways to use that information, especially for environmental testing, to inform decision-making at the hospital level, to make the operation of the hospital more efficient, but also to help clinicians to make decisions that will ultimately drive quality of care. One clear example, for instance, is antimicrobial resistance, right? Today, the antibiograms that doctors use are great, right? But oftentimes they don't really rely on recent information or information that is specific to the hospital. What if we could provide an antibiogram to the hospital based on the wastewater testing in the hospital? So that's what we're launching right now with a number of hospitals in the US. In this specific slide, we're talking about some of the work that we've done with digital surveillance. So taking data feeds from other programs and showing them to the hospital in a structured way. But I think really the real power will come when we're gonna add those environmental surveillance data feeds. Another example is, so I was telling you about the airport program in the US. Well, the reality is that the concept has been very successful, and now it's been replicated across the world, which makes sense because that is the basis to build a global network. So this is an example in Rwanda, where Kigali Airport has replicated a very similar program to the one in the US. They are also testing aircraft and passengers in a very similar way. And the cool thing about that is that they get a coverage of the region in a way that the US wouldn't get. So that's when you start really seeing the power of a network. The gaps for one country are covered by the other country. And if these two countries share data, they can achieve full coverage of essentially the world. Let's see what else. Okay, last example here is a program that we're doing in the US. I cannot say the city because we haven't announced it yet publicly, but essentially here we're taking this approach of community health holistically, and we're testing, in this case, not for pathogens. In fact, somebody else is doing pathogens. We're doing the high-risk substance piece of the equation, so that the public health agency has a really holistic perspective on the health of the community. We're doing this by neighborhoods, so we're able to tell the public health agency where the concentration is highest and where it's lower, and that ultimately is supposed to help the public health authorities, and especially the recovery services, to direct their resources in the most efficient way. Otherwise, oftentimes they have to rely on very, I would say, on back-looking, if you will, data, right? Essentially overdose cases and fatalities, but that's essentially a lagging indicator. When that happens, it's too late. There's very little you can do. What if you can get a forward indicator, something that tells you about the future? And that's where wastewater comes into play. It can be this kind of forward indicator that will tell you about a potential increase in cases that will happen in the future, in the short future. That's it for my presentation. I hope it wasn't too verbose. And again, thank you so much for following. Am I okay, Alex, with time? Was I too long?
No, perfect.
That's perfect. Yeah.
Okay, good.
Yeah, thank you, Robert, for the great talk. And it's amazing to witness how rapid growth and development of this field since the onset of the COVID, and especially seeing those achievements made by Ginkgo. And I really appreciate your sharing both the fancy part and the tricky part as well. Yeah. And okay, for the audience, please feel free to share any comments or questions you have in the chat box, and we will address them later after our panel discussion. So without further delay, let's extend a warm welcome to our second speaker, who has worked extensively with the first-hand data in the lab during the COVID time. Shu-Yu, the stage is yours.
Thank you. Okay, just making sure everybody can see my slides here. All good?
Good.
All right, so here today I'm gonna introduce the Missouri SARS-CoV-2 wastewater surveillance. So this project was launched around 2020 May, something like that. So that's like right after lockdown, and just touch on the basic stuff. So what exactly is the wastewater-based epidemiology? So basically, we're monitoring the pathogens or drugs in the population by analyzing the wastewater, and as Robert already demonstrated, it could be simplified as sample collections, analyzed for the indicators such as gene markers or chemical markers, and at the end we perform the data analysis for like public health decision. And then here comes the question: why do we use wastewater-based epidemiology? So first thing is it's a near real-time monitoring for early warning, as Robert already mentioned like a couple of times in his talk. So and then the other important thing is the wastewater-based epidemiology can capture the asymptomatic infection, because humans start shedding the virus through the GI tract even before they show the symptoms. So in this case, if we could monitor the wastewater, we could start capturing the infection as early as possible. And compared to the individual testing for larger scale at a community level, it's relatively cheaper. And we kind of did a rough calculation. Every single sample for one sewer shape probably only costs like $2 per round for depends on like the frequency you run, but it's pretty much like $2 per sample in this case. No, $2 per person for that community. Sorry, my bad. So on the right here, this is the map of Missouri. I know a lot of you guys don't know where Missouri is. It's middle of the US. Yeah, it's really like a really square area. So all the dots here, orange or square, are the sewer system that we monitor. So we cover more than 75% of the Missouri population in this case. We are the largest wastewater surveillance project across the state, like across the United States. So in the beginning, when we start looking at SARS-CoV-2 in the wastewater, we have a lot of uncertainties about it. Is it still infected? Can we safely handle it? So we did some experiments to study and characterize the raw SARS-CoV-2 in the wastewater. And we understand that like the SARS-CoV-2 particle is an intact envelope particles instead of just the loose mRNA in the wastewater. And second, we also try to understand what's the best way to transport the wastewater samples from the facility to our lab. So we do the pasteurization and freeze-thawing cycles to see how the SARS-CoV-2 signal behave. And we found that going through those cycles can greatly decrease the signal on the qPCR assay. And at the end, we figure out 4 degrees is actually the best temperature for transportation. And that kind of coincides with the mRNA characterization as well. If you have worked on RNA samples in the past, you might know that going through the freeze-thawing cycles can degrade your mRNA quickly. And lastly, we also did the inoculation of the wastewater on human cell line, and we did not find any infection symptoms on the cell lines. So we are confident that we definitely can, quantify and characterize the SARS-CoV-2 in the wastewater. However, there's still some challenges. First thing is the scale-up process. So in the beginning, in our pilot study, we only did about like 25 samples per week. But in order to cover the 75% population in the state of Missouri, we actually need to analyze more than 100 samples per week. So that we have to optimize the process of the RNA extraction and also the qPCR assays. And during pandemic, I think everybody has been through this like there's always an emerging SARS-CoV-2 variant, and we always want to make sure that like which variant is prevalent right now. And something interesting we found out throughout over the time is that we see good correlation between the confirmed case number and the concentration of SARS-CoV-2 in the wastewater. However, if we look at individual sewer shade or facility, we sometimes find out there's no good correlation or proof correlation in that case. So we hypothesized maybe it's the lack of population biomarker that was affected by the dilution of the water going to the wastewater facility. And the second thing is some chemicals in the wastewater could affect the behavior of the SARS-CoV-2 particles. So the first thing we did in order to combat those challenges is to improve the efficiency using the automation. So first, we bought the qubit that will help us to do the RNA extraction without any contamination. And we also use robots to help us to load the plates for the RT-qPCR assays. And our turnover time is within 24 hours since we received the sample. And then later on, we also we have been adaptive to the emerging of the SARS-CoV-2 variants. So we have to constantly redesign our primers and probes in order to capture that mutation. And as you can see here, this is just a screenshots from our website. So you could see on the right here, that's a percentage of different variants over time. And then I mentioned that earlier, we need the population biomarker for properly interpret the prevalence of SARS-CoV-2. So the prevalence is defined by the viral load divided by population. And we were looking at several populations, including Pepper-Mild model virus, abbreviated as PMMOV, that's recommended by the CDC in the very beginning. CDC kind of assumed that everybody eats pepper, and pepper contains that virus, and that virus can be found in the stool. And then we also monitor creatinine, which is the muscle metabolites, and 5-HIAA, which is serotonin metabolites. And we also have the other two biomarkers might surprise you guys. That's the caffeine and caffeine metabolites, para, same thing. So the reason why we monitor caffeine and caffeine metabolites is, I guess, everybody drink caffeinated drink at some point. If you don't drink coffee, you probably is a tea lover. If you're not a tea lover, you probably drink soda, especially Coke or Mountain Dew, you know, like all those sodas or drinks, beverage contains certain amount of caffeine. And as we suspected, we perform the correlation analysis between the biomarker loads versus the population from the census. And the caffeine and caffeine metabolites won't. They have a really good correlation, R squared equal to like 0.93 or 0.94. So in one of our study, we actually used this population estimated, biomarker estimated population to do the normalization of the SARS-CoV-2 in order to see a better correlation with the COVID-19 confirmed cases. And the other thing is the compounds that suppress the SARS-CoV-2 in the wastewater. So we found an interesting result by looking at, here is all the sewer shades we monitor or wastewater facilities. And then supposedly the copy number divided by the patient should remain the same for all the facilities. However, we see different ratios at different facilities. So it got us thinking there must be something that suppressed the SARS-CoV-2. So we did the global profiling using metabolomic approach, and also the statistic approach to identify the compounds. And we spiked those compounds in the wastewater that contains SARS-CoV-2. And as you can see on the right, those two are the example compounds. They actually suppress the signal throughout the time. So now, since we are in the post-pandemic era right now, like what other things we are doing for this project? So that includes a lot of still genetic biomarkers for Mpox, influenza A, influenza B, and like Robert mentioned, RSV is kind of hot topic right now. And then for the chemical biomarkers, now we are still quantifying the caffeine in the wastewater because it reflects the population. And when there's a flood going into the wastewater facility, they cannot calculate their flow rate at all. So we actually use a statistic approach to back calculate what's the flow volume that going to the wastewater facility. And the other thing is we are actually monitoring the opioids because CDC has declared the opioids epidemic right now. So that's also a heavy focus for this project. And just some fun facts. Robert showed how the samples are collected, and then I don't know if you guys were wondering, but when I first joined this project, I was wondering like how much sample volume do you need in order to analyze and tracking the SARS-CoV-2? And the answer is 50 mil. It's not that much. And then for the chemical analysis, sometimes it could even go down to like 2 to 5 mils, depends on your extraction protocol. And the other thing is how many people are on the University of Missouri team. So for us, it's like 20 to 30 people, always on site pretty much. And then half of the team are focusing on bioanalytical analysis, where I was in before. And then half of the people are working on RNA extraction and sequencing. And I just like to thank my advisor, Dr. Chong Ho Ling, and then my mentor, Dr. Xin Yeshef, for supporting the project and leading the project, and our partner, Dr. Mark Johnson, and also our sponsor, the Missouri DHSS, Missouri Natural Resources, and University of Missouri. Yeah, and if you guys are interested looking at the wastewater surveillance websites, that's a QR code on the left, and then the QR code on the right will link to my advisor's webpage. Yeah.
Great.
Thank you.
Thank you, Shu-Yu, for the nice talk. Yeah. I'm impressed how you simplify the complexity of the project and how your team tackle the challenges. And I personally feel that it's really smart to use pyrazintine from caffeine as the population biomarkers. Thanks again for these two excellent presentations. And it's really great to have a large overview of the current landscape in biosecurity and also the pathway of the pathogen surveillance. And so now let's go more detail to the projects that Robert and Shu-Yu are working on through the discussion. For the audience and also like Robert, Shu-Yu, if you both have questions for each other, feel free to chap in at any time. Okay, so my first question is what opportunities or inspiration led to the original of your respective program? And can you elaborate more on how your program got started? Shu-Yu, would you like to go first?
Yeah, sure. So like I mentioned earlier, like during the lockdown, we were just like staying at home, get bored, and at some point we were looking at some literature, and some publication indicate that SARS-CoV-2 shared it through GI tract. So that will end up in the whole stool. So my advisor had a crazy idea, like, why not using wastewater, since that's where our poop are going to, right?
Yeah.
So that kind of is the inspiration for our project. And honestly, our partner, Dr. Mark Johnson, he was skeptical about this. He's like, it's not gonna work, because he's a virologist. He's like, no way you could detect it in the wastewater, but it worked, and it worked beautifully.
Nice. How about you, Robert? I know you're already sure how you started that.
Yeah, I mean, for us as a private sector company, public health is not necessarily something that we were doing before the pandemic, but with the pandemic, we realized that there was a need. Like there was just a need for more scale, more support. There was a need for the private sector to bring in their, way of thinking about things in a cost-efficient, scalable way. And so we were very successful during the pandemic, and then we really worked hard to pivot our team so that we could be still relevant in a post-pandemic world. And I think, in the beginning, Alex mentioned how it is important to have this sort of infrastructure to prevent the next pandemic. But I also think that it can be valuable in the inter-pandemic time as a way to learn more about the health of a community and be able to, trigger the right response action. So is not just about pandemic prevention. It is, obviously, but it's also about delivering value in the long term. So the reason why we are involved in this is that we really believe that the private sector has a role to play. Obviously, funding can be tricky. We can talk about it, but ultimately, we believe that it's just a matter of time until this is going to be a really big thing, and potentially also a market, an attractive market for the private sector.
Thank you for sharing, and it's truly meaningful start. And yeah, we are really lucky to have like people of you like you to work on these fields. Yeah. Okay, so for my next question is, from the beginning of the program to now, can you share with us a successful story that were results from your program, maybe causing a policy change or establishing countermeasures, or also mitigating some public health risk? Robert, would you like to take the question first?
Yeah, so a lot of the things that we do are not public because we work for clients. So I cannot really disclose all the details. Some of it I was able to disclose it earlier, so we were pretty early in the detection of some of these variants. What does it mean to be early? Well, it actually means that we can provide a number of information to our clients, to our public health partners. First of all, we can provide genomes. And from the genome, it is possible to infer how the pathogen will impact the community. Things like relative growth, things like immune escape can be inferred from the genome itself. But also the samples can then be cultured, and that culture can then inform again a lot of important information about the pathogen. Another example that we have been able to contribute to was the fact that some time ago there was a scare around a new pathogen, and especially around the resistance of that pathogen. And so in one of our programs we're able to get a sample that was positive to that specific pathogen, and we were able to essentially assess the resistance to a specific antibiotic. In a way, that exercise influenced then the policymakers in the sense that if without that information, maybe they would have potentially closed the borders, or they could have started some other controlling mechanism. But because the sample that we were seeing did not have that resistance, in a way it calmed down a little bit the environment. So this is not just about triggering also response mechanisms, it's also about not triggering them, right? If you actually realize that the risk is not as high as you thought, sometimes the decision is not to act, which in many cases can be a good decision, given that acting oftentimes carries a very big economic, social, human cost. And so sometimes the non-signal is just as good as the signal, because it enables you to make a more informed decision.
Yeah, great. It definitely has lots of challenge in the program. Good to know that how to address the problem. Okay, thank you. And Shu-Yu, can you share one story that impressed you the most as well?
Yeah. So a few of our storage samples are from correction center and elder homes. And then honestly, elder homes are like they have the most vulnerable population over there. So one time there's an outbreak. I think it's all during the Omicron variants prevalence, and then we knew one of the elder home has like really high concentration of Omicrons, and we immediately informed them, and then they started doing the individual testing for all the elders in that facility, and they could isolate it in like them in order to prevent the further spreading. So that's something like I see like our impact, and also like in a positive way. So yeah, that's really like inspiring.
Great. Awesome to hear that. And thank you for providing a concrete example for the real world impact that's happening around us. And when talking about the current technology, these questions also from the audience in the chat box, and it seems to be adaptable when we have prior knowledge of the threat, right? So I believe many of us are really curious about how you determine what to measure in the absence of information about the future potential pandemics. Shu-Yu, can you share with us how people working in lab addressing this uncertainty?
In our lab, we don't do it, to be honest.
Yeah.
But it really depends on the capacity of the research center or institute. Because for my understanding, if you want to monitor something is unknown, that means you need to eliminate the known first.
Mm.
So oftentimes you have to do the whole genome sequence of your wastewater sample, and then you have to use like a large computational power to eliminate all of your known sequence by aligning them with all the pathogens. Then you might have something that's out of ordinary. Then you will start looking into that. So there's a lot of like computation and computational power that's required, and also the facility capacity. So now I think not a lot of institutes or like even private entities are doing this, but I did hear one company are trying to heading towards that direction.
Mhm. Okay, thanks for Robert. Would you like to share your thoughts on this?
Yeah, absolutely. So that's honestly what we are currently moving towards. It's called metagenomics or pathogen agnostic characterization and testing, and really it's ultimately what our clients are asking for, right? Yes, of course, SARS-CoV-2 is still as important and interesting, but we want to get to, first of all, to a very large set of targets. And so
Yeah.
there's a number of, for instance, target enrichment sequencing methodology that they kind of go around the problem that Shu-Yu was mentioning, right? They solve for it because instead of essentially amplifying everything, they just focus on specific targets. But by doing so, you can still get information about hundreds of different targets. So that's kind of the intermediate step. And then you have the full pathogen target agnostic sequencing and metagenomic, and that's really ultimately the goal. But like Shu-Yu was saying, the issue there is that signal to noise problem, right? Where you have a lot of things that you don't really care about. So we are addressing that with a number of technology. One is called depletion sequencing, where we are essentially removing from the sample all the things that we already know about and we're not interested, and really focusing just on the new sequences, on new genomes. It's feasible, and to be honest, we have the technology today to do it. The problem, as Shu-Yu said, is that it requires a lot of compute power, it's taking time, and the goal ultimately is to make this as quick as possible, because you want to get your result quickly. You don't want to wait days or weeks before getting a result. So what we're working now is on essentially point of collection, point of testing devices that will be able to give you that kind of metagenomic, or at least very large, panel information on the spot and within minutes, and not hours or even days. So that's part of some of the development projects that we're doing in the US, in Europe. It's what our clients are asking for. And I think honestly, the technology is there, just about bringing it down to the parameters that make it usable and practical.
Yeah, it makes sense, and like seems promising to me. Yeah. And thank you for sharing your expertise and delving deeper into the technology challenges. And Alex, would you like to drop in with different aspect?
Yeah, sure. I'll take over the discussion for the bottom half of this panel. So one thing when I approach this topic, the first thing that sort of jumps out at me is really about the data that's being generated in both of your program. Like, in addition to the biological data that, where's the virus and what variants are the virus and all that stuff, I think there are a lot more metadata. So this is a very enriched set of data that it not only tells about the biological, but also tells about the behavior of the travelers or the elderly inmates and so on. So I think it's a very powerful data set. So my first question is, so who owns this? Like how do we deal with data ownership in a program like yours? And also, are there privacy or ethical or other sort of concerns or pitfalls that you might be looking for when you're discussing this topic? Why don't we start with Shu-Yu on this one?
Yeah, so for our project, the state of Missouri owns the data. So that's kind of obvious because we report all the data to them. But the University of Missouri also have the ownership of data, which means we could we could manipulate it, not really like, not like we could use it for like data analysis and then trying to use them for publication in that case. But the announcement or the data like released to the public is through the state of Missouri, it's yes, through the state instead of through university. We could only do the publication in that case. And as for the sample wise, sample is also owned by both institutes, so both university and also the state. However, majority of the samples are stored at our institute, like at University of Missouri, just because we do have the capacity for the storage. And then we do have agreement that we could still use those samples to do further like experiments, because the funding is from NIH. We have to generate certain kind of report to them. So there's a mutual agreement between the university and the state, but it could be different for different like projects in this case.
And Robert, would you like to comment on how your case is handling this situation?
Yeah, I would say it's very similar. For us, it's very important that ultimately the public health authority owns whatever data and samples that we produce. We do help them to analyze them, so we have some sort of like license so that we can still manipulate the data and use it. The important thing to remember though is that this data doesn't contain any human genome, because any human genome genetic information is filtered out. And so it's really only about pathogens, right? And ultimately we're working for the public health authority, so the goal of all of this is the greater good. The goal of all of this is to help, right? And so that's something that we are very careful about. Besides that, our company, and you know this, Alex, has a specific team called the Caring Committee. It's a committee of selected people who always make sure that anything that we do from a research and development, or even from a program standpoint, gets discussed using an ethical lens that is, to be honest, very, very thorough. And so we're not just like ultimately serving public health, and so in a way those decisions have already been pre-vetted by the public health authorities, but also within ourselves we have a mechanism to check that everything we do is ethical, and so from that standpoint, I would say it's an additional guarantee that we are really making sure that we're taking into account every possible angle and every possible like concern around data ownership and use of that data.
So
and
just
I want to, oh, sorry.
go ahead.
I just want to add on one thing. Like when I was working on the project, I also had a concern about privacy, because everybody's stool goes into the wastewater, right? And like Robert mentioned, like Ginkgo might filter out the human genome, but for us, everything is still condensed in the sample. Like, so I did have that question before, and I talked to my advisor. Like the answer I got was that we are not looking at the individual building. We are looking at the community level. So it's not, we don't look at the information into one person only. So that doesn't consider as invasion of privacy, which kind of makes sense to me. Yeah.
Yes, good point.
Yeah, so sort of going along this track of data. So like this is, again, just going back to the idea that this is a very unique set of real life data of like real people in the world. And so not to like being too attached to buzzwords, but what do you think about incorporation of machine learning? And with this kind of data, so what kind of applications or future vision can you see by combining these tracks? Robert, do you have some thought on this?
I'm very excited about the use of machine learning and artificial intelligence with what we do. Ultimately, we're working with massive amount of data. For instance, when we were talking about metagenomics, right? We're talking about huge data sets, and right now a lot of that analysis is done by people. And as many of you know, bioinformaticians are probably some of the people that are the hardest and rarest to find. So there's immediately like an efficiency opportunity here to reduce the manual work and make that work being done by machines, right? Secondly, I think one big question that we still have is the prediction genome to phenotype. In other words, we know about genomes. We can read the genome at this point. We have really good technology, but it's very hard still to predict what the phenotypical response is going to be, right, from that genome, because we know that it's not one gene, one specific phenotype. We know that the relationship is much more complex, and so I'm really hoping that this kind of technology will help us to get better at predicting that relationship, especially because the data set are growing, right? We have so much more data today than we had in the past that we could potentially use to correlate the genotype to the phenotype. And the third piece is moving a little bit away from genetic information. What our team is also doing is using a lot of non-traditional public health data to predict outbreaks and predict how an outbreak is going to evolve. And they're using information such as mobility, mobile phone information, school attendance, and all of that kind of non-traditional data set. And the goal is to be able to predict future outbreak. We're actually doing one of these projects is with the Gates Foundation. They are funding us to do some of this work. And in this case, it's about predicting where measles outbreaks are going to happen, right? So big issue, especially in low and middle income countries, where, by the way, there's also very little availability of data about outbreaks and cases, but there's other data available. And so we're hoping to use artificial intelligence to sift through all of this data and be able to make this kind of prediction. So I'm very excited. We're definitely, Ginkgo is very invested, as you know, Alex, in artificial intelligence. We have a partnership with Google, and we really believe that AI will unlock a lot of the power and opportunity of biology.
Yeah, no, absolutely agree. And Shu-Yu, do you have more to add to this?
No, I'm not an expert of AI. But yeah, I agree with what Robert said. I think that's the future, and that could help with like filtering out all the like known stuff instead like to get to the unknown stuff in this case. Yeah.
Yeah, so change track a little bit. So one of the reasons why I'm excited to assemble this panel together is I think we are getting basically a very similar type of program in the pathogen detection, but coming from two very different sides. One, Robert is more from a private entity, and Shu-Yu is more from an academic institute type of studies there. So I'm just curious about both your thoughts on the roles of academia and private entity of working in the same space, and how can each side contribute uniquely in their own way. And would Shu-Yu like to start first?
I think I would say educational institute does have more freedom regarding the research part, especially when we partner with the states or regulatory authorities. I do feel like we do have more wiggle room that we could play with the samples. So in this case, I would see like educational institute as like a powerhouse for the knowledge or creativity in that case. But to some point, if it has to be scaled up or you have to be like more compliance with the regulation in that case, like for ethical reason, for like for regulation wise, then it's probably good to partner with the private entity or private sectors because they know the compliance of the regulatory better than us. For us, we are like for the scientists in the academia, we like to explore things, but at some point, I feel like if we want to, for mass production wise or for scale up wise, it's good to let the experts like help. Yeah.
And Robert?
Yeah, so I would say that the lines between the private sector and academia are much more blurry than they used to be. Ginkgo was founded by 4 scientists, actually 5, including obviously Professor Tom Knight. And in many ways, Ginkgo is an academic institution because we do a lot of research, but obviously we also have an angle towards scale, like Shu-Yu said, and ultimately commercialization. So the lines are very blurry, and universities do the same, right? They have now a lot of programs to make sure the technology and the research gets then used to launch businesses. So that line to me is much more blurry today than it's ever been. And that said, it is true that there is a little bit of a slight difference in mindset. We are very focused, and especially myself, that's kind of what I like really to do, is to focus. I focus on the use case, right? What is the use case and what is the ultimate value to the user? And so in many ways, I leverage the research that academia does, and I try to go one step further, and I ask myself, okay, who is going to benefit from this first? And how can we show the value? And ultimately, how can we monetize the value? And if you think about surveillance, right? Everybody says surveillance is helpful. Everybody. There's nobody, I mean, some people will say that, but the majority instinctively understand that this is important. However, when you ask them to pay for it, that's when people say, well, actually, we have no money, right? So the focus on finding the use case and making sure the value can be monetized is more important than ever. If we don't do that, right? If we keep telling people wastewater is useful and we show really good research, but we cannot link it to actual value, unfortunately, we're going to be again victim of the cycle of pandemic. The pandemic neglects that happens after the moment of like crisis, and so for me and for our team, this is so important. It's not just an endeavor driven by the fact that we are a for-profit company. It's mostly about finding a sustainable model for all of this work, for all of this technology to continue to be used and in fact get better as opposed to be abandoned. And really, we're talking about something that is happening now, right? Right now, if you look at the number of sequences for SARS-CoV-2, they're plummeting. There used to be thousands of sequences every day. Now, all of those public databases are getting just a few hundreds, if they're lucky, per week. And that's going to get even worse. However, we do have a window of opportunity, and that's what we're really trying really hard at Ginkgo. We're trying to leverage that window of opportunity, find a sustainable model so that all of this infrastructure doesn't go to waste.
Great. No, I absolutely agree. And I think that it's also very important that both sides contribute in its own way, but at the end of the day, it's what really benefits and what brings these technology into a field that can create these practical and beneficial effects and ultimately impacting the society. That's the more important thing here, especially for the kind of services that these programs serve for. So in the interest of time, I will pitch my final question for the panel. I think it's a very exciting, for me personally, it's a very exciting conversation. I wish we could just go on, but timing and all that. What do you think as involvement in a program? Do you see it as the future vision of these programs more in like short term or long term? Like, where do you see this go? Do you see like sustained support from the government to keep this as a long term thing, or do you see it more like fade away as other pandemic related technology goes on, or what do you like to see from your respective program, how to evolve into maybe a 5-year, 10-year, into the future? Robert, do you want to start us off with this?
Yeah, absolutely. So honestly, that's the focus of what I do every day. It's finding ways to make those programs relevant at the micro level, right? To the hospital, to the factory, to the airport. So I want that data to be very useful at that level to drive efficiency, to drive quality of care in a hospital, but also I want that data to be then to be used at the macro level by public health to exactly perform that kind of pandemic preparedness and response. So if we can find that right balance between the micro and the macro, if we can find the right balance between the more utilitarian use case of that data, but also the public health use case, then I think we have created a sustainable model. And that's really what I hope for the coming years, is that we can find a sustainable model. Obviously, the technology has to get better and cheaper and faster, but ultimately it's about finding use cases, right? It's about finding value beyond public health, value for industry, value for agriculture, value for healthcare. I hope that the pharma industry will also step in. There's a lot of conversations right now whether pharma should be part of surveillance. I think they should be. I think they should be part of the conversation. So I hope in the next few years we'll find that there's a lot of stakeholders that have can drive value from these technologies.
And how about Shu-Yu? What's your thought on the future of the wastewater monitoring program?
So I would say I would stand from like academia point of view, and then I hope that like in the future, wastewater-based surveillance is not only for pathogens or like any kind of emerging, viruses or any kind of like microorganism. I wish in the long term it could also be used for monitoring like peptide biomarkers, which means you could even quantify how much antibiotic is produced in the waste and then released into the wastewater that could against those pathogens. So that gives us another layer of information, not only the prevalence of the virus, also the immunity of the community. So in this case, we would know if this community is strong enough to fight against the pathogen or not. If they are, then the delegation or like the mitigation could be put into other places rather than that community. So that's something like I hope to see. However, like peptide analysis in wastewater is more complicated than just nucleic acids. But I do hope like with the advance of technology, we will see that happen like soon. Yeah.
Great. I think that's a fantastic place to wrap up the panel discussion, as I hope for this hour and some both presentation and as well as the panel discussion, the audience, at least for myself, I know I've learned a lot, and I really appreciate the effort that's been contributing from both program and really the importance of how these programs impact our lives. And as well as I just hope that I give some opportunity for pandemic-related technology that's not vaccine or therapeutic-related to a chance to shine a spotlight, as they are also super important, especially in the post-pandemic world, to really help us really protect against the next wave, and also other sort of application, as Robert and Shu-Yu has described during their presentation. So with that, I'd like to close out on the panel discussion section and move on to the audience Q&A section. And our audience has been great tonight. We have a great lineup of questions in the chat box already. And so we will try to go through those first. And if anyone has any questions, feel free to continue leaving in a comment or raise your hand, and we can address them accordingly. So first question is from Chief Wu Wei, and I will read a question. Great talks. What would you suggest for regular monitoring in wastewater? And I wish to the priority based on costs. I think efficiency consideration. So I think it's mostly about the cost involved in these, and what would be the consideration there if cost is being factored in. So I guess it's more relevant to Robert.
Sure. Absolutely. Cost efficiency has to be a consideration, because if we had unlimited money, we would put, pathogen monitoring programs everywhere, and not just wastewater. I mean, you would also have swab everybody's noses, and you would have air monitors everywhere, for sure, right? In reality, we have to be more strategic. We have to think, what are those nodes where you're more likely to find something interesting? And so that was a little bit part of the presentation I gave in the beginning, right? Talking about the strategic nodes. Airports is one, because that's where so many people from all over the world are going to be congregating at some point and passing by. Mass gatherings are another one, where you have people from all over the world coming together. Farms is an interesting one, where you have kind of the wild environment and the human environment coming together. And that's where zoonotic effects can happen. I mean, let's remember that the majority of infectious diseases actually come from zoonotic spillovers, right? So if you can identify those nodes, then that is where it makes the most sense to position those detectors. The other argument here is to think about where are those places where there's an interest there's an economic advantage in having monitoring systems. Farms, again, is one where you could imagine having a monitoring system that monitors for pathogens that have an effect on the yield of the farm, right? So you could monitor for things like coccidiosis at the farm level, but at the same time, you could use the same sample for monitor for HPAI, right? So let's think of a situation where there is a win-win, where the data can be used for, productivity purposes to drive productivity or other things like quality of care, but also can drive then information about public health and pandemic preparedness. So that's what we're doing essentially in my unit. That's the kind of things that we're thinking about.
On a related note, I just have this tangent off my head that I thought is appropriate to put in here. So in your case study, one of the example you show is airport monitoring in Africa. So as you mentioned earlier in the presentation, that money is a big thing. So now you ask people to pay up. Some people start to hesitant about having the program. So how can we be sure that even though money is a factor, how can all the countries around the world, whether it's the developed country like US, Canada, or underdeveloped country like in Africa or other places, all gets access to this kind of technology?
Yeah, so that's actually one thing to dispel, right? Those programs don't have to be very expensive, right? Obviously, when they are in research phase, they are expensive because we're trying to figure out how to do things. But as we move down the technology curve, there's a lot of things that can be made more efficient. We weren't talking about, for instance, bioinformatics, right? If you do bioinformatics in a very manual way, then you're going to need a lot of resources. It's going to take time. But if you can automate as much as possible from those pipelines, then all of a sudden it essentially becomes the cost of computing, right? But you have minimal labor costs and so on. So there's ways we can make that more efficient, and that's what we're trying to do at Ginkgo. Ultimately, however, I would say those countries may have fewer resources, but they still value the security and safety of their people. They know what the impact of those pathogens can be on their population. In fact, in many ways, they've been more exposed than anyone else to these pathogens. So there's definitely a willingness from the governments to invest in those technologies. So paradoxically, we have found a lot of interest in regions like in Africa in those programs. And let's not forget that there's a lot of funding mechanisms to fund these activities, right? So there is money available. Obviously, they want to see that it's being used correctly. But I think what Ginkgo is doing is that it's really, because we are enabling a lot of cost efficiency, we offer services that are very attractive to those countries because they can actually get a lot of value out of those funding that they receive from those like global multilateral mechanisms. So it's obviously a question that we're still trying to answer fully, but definitely the idea that surveillance is something that people are not willing to pay for, it's just not true. There is a lot of interest right now.
Great, thanks. Let's move on to the next question from Jerry Shea. I think the first question has already been addressed earlier in the presentation, so we'll hop to question number 2. It seems like we are testing the things the question number 2 has been addressed. We'll answer question number 1. It seems the surveillance depends heavily on cooperation and consent from government or the property owner. Will this be a limitation of the business model, or if there is a possible project with private sector? Who would like to take this? Shu, would you want to comment on this?
So when I was fresh out of PhD, I actually looked it up like different companies for like jobs. I do feel like there are more and more companies that are open to wastewater surveillance, and then Ginkgo is one, and then Verily, which is under Google, that's also the other one. So at least like there, and then I would say some of the institutions, they do have their own research center that's not really directly like governed by the university wise. They also start doing those wastewater surveillance. So I would say there are more and more. Just depends on like the region and then what kind of applications they're using it for.
Great. Thanks.
Maybe Alex, if I can add a little bit here, it is true that to get samples, you have to get a consent of the donor, right? So one example is aircraft. We really think that it's super powerful to collect samples of wastewater from an aircraft, but not all the airlines want to do that for a number of reasons. And usually it's a perception, because the truth is the sample is anonymous, and we can't really say anything about, but still some airlines are uncomfortable with that. So what we do is that we find ways around it. So one thing that we do is that when we cannot collect from the plane, we're going to collect from the triturator, which is the place in the airport where all the wastewater gets dumped from the planes, right? So it is a bit of a diluted sample. We get more signal because it's not going to be just one plane, it's going to be multiple planes, but we can still get valuable information. So it's also up to us to be smart, and find ways to still get valuable data without having to, without getting blocked by stakeholders that may not want to collaborate. But ultimately, I think it's also a mindset, and it's just over time, people will understand that this is for the greater good, and there's no risk for them as an individual company or facility. And so I really think that over time, we will see more and more people participating and consenting to donate samples. But it's true that today there's still some resistance, and it's up to us to be smart and find ways to still get the valuable data that we need.
Great. That's a great follow-up. Thank you, Robert. For next question is from Director Tsai. Thank you for your wonderful talk. I have a question that since the virus can be detected from wastewater, should the state government further treat or clean the water to prevent further contamination? Shu-Yu, do you know anything about the follow-up here?
So, yeah. So basically, our samples is the raw inlet from the wastewater, so it's untreated. And then a lot of paper has shown that once they are treated, they are no longer in the wastewater. You barely can because the viral particle, the envelope, will be destroyed throughout the wastewater treatment process. And then it's likely that you might still the mRNA fragments could still be detected, but they are no longer infectious. So yeah, if that addressed the question. Yeah.
No, great. No, that definitely provides some clarification and address some public concerns.
Maybe adding one thing here, Alex, for the wastewater, it's true that especially at the level of the wastewater treatment plant, we're measuring the influent, but the effluent goes through a process of I would say treatment and cleaning. So the risk is lower. But I'm thinking of a different situation, building and air in building, right? So there is more and more a realization that the quality of the air in a building can really impact the quality of the population that is using that building, like schools, for instance, right? Or hospitals. So there is now, it's not wastewater, I know, but it's a similar idea, right? There is a focus on developing solutions that can detect pathogens in the air and then modify the treatment of that air through the HVAC system so that we can neutralize any sort of pathogen in suspension. So this idea of a closed loop system where there is a sensor that then can activate some sort of like response to clean the environment is definitely top of mind. Ginkgo is working on some of those technologies. And I think, again, in a few years, it will be normal to have that kind of detection in every building where there is a risk of contamination. Again, hospital and schools, nursing homes could be another one. So just to provide some more color.
No, I think that's also a great segue into the next question from Li Yu. Thanks for the talk. From the talk, I guess the sample are mostly analyzed in the lab. Is there also an attempt to do the sampling, analyzing, and upload in the field, like we collect air quality or temperature data? Robert, would you like to fill in any gaps here or
100%. I mean, that's exactly the next step, right? The idea that you can collect a sample and then ship it to a lab and it takes 48 hours and even longer for sequencing, it's just not feasible in the long term. We know that the signal has to be, especially if you want that signal to be useful at the local level, at the micro level, it has to provide a result right away. So there's a number of technology already that do some of that, but typically they're not multiplex. They only provide results for 1 or 2 pathogens. We are working actively to get to a much bigger multiplex, so like 100 targets. It's really going to happen. I think it's just a matter of technology. It's not whether the science is feasible. The science shows that it is feasible. It's just about finding the right technology, and I really think that that's going to be pretty ubiquitous in the future.
Great. And I think in the interest of time, we'll take one final question from the audience. This one from Dwayne. Since the later stage of the COVID-19 pandemic, people have increasingly shifted from centralized PCR testing to the point-of-care antigen tests. Has this presented a challenge for surveillance effort? And if so, do you think there might be solutions to crowdsource surveillance data, for example, blockchains for biosurveillance, and provide incentives such as Bitcoin mining? So this multi-layer question. Robert, would you like to start on this?
Yeah, for sure. It's actually very true, right? That for instance, I remember in the UK, they were one of the first countries that shifted from PCR-based to antigen-based testing. And from almost like overnight, the entire data that they were collecting went away, right? They had no more data about the pandemic, which was a big issue from a public health standpoint, because they didn't know what decisions to make. So there was obviously the advantage of getting that results immediately and everywhere, perhaps with less accuracy, but ultimately there was an issue with the overall reporting. So what we do in our program is that we offer the option for users to send a sample to the lab if they test positive. So the idea is that you're going to use your antigen. If it comes back positive, you can send a test to the lab for further characterization. We'll tell you what sequence you had, what lineage you had, and so on. So we did that during the pandemic. Obviously, we still rely on the user to make that extra step of sending a sample to the laboratory. As we move forward, the point of care devices that we're developing will have embedded a way to upload data to the cloud. And so in the future, obviously, antigen maybe not, but if you're looking at those multiplex devices that will be able to tell you in real time what results you have, the idea is that those devices will all upload the data to the cloud and will eventually go into a big database that will enable us then to do that kind of analysis at the macro level. So that's going to be a really important thing to do. The incentive there is ultimately, first of all, we could build this into the machine, but the incentive could be that ultimately, if you do that, the user may also get data from other nodes, right? So we have to think of this also as a data sharing system. Is there a way for the user to upload the data, but also get down data that would enable them then to compare their results with what happens in the community and be able to do some more contextualization of the data? So an imperfect answer, I guess, but that's, again, something that we're definitely thinking about right now.
Very fantastic. Thank you so much. And I think with that, we will close out tonight's event. I just would like to, again, thank you for both Robert and Shu-Yu for spending the time with us tonight and able to share all their experience and expertise. And I know for myself, I have learned a lot in this process, and I hope all the audience online have as well. And so with that, again, thank you, Robert, Shu-Yu, and again, thank you to my great co-host Chi and Alex and Chi here, signing off. See you next time.
Thank you so much. Thank you to the moderators.
Yeah,
Fantastic
thank
job.
you.
Thank you, Shu-Yu.
Thank
Nice
you
to
all.
meet you.
Yeah, nice to meet you.
Yeah, you're both definitely the distinguished representative in this
Did
field.
we meet the bar? Was it worth it to stay
I believe
up until
that.
9:30? We've earned a drink or a tea or whatever you guys like to drink at
Yeah,
night.
I already have my tea here.
You start already, Chi.
Yeah.
I still got some of those like Ginkgo ciders in the fridge, so I gotta have one of that after.
Are you still in the office, Alex?
No,
Oh
no,
my
I'm at
God.
home. I'm at home.
Oh,
Yeah.
no, okay. For a second, I thought you were still in the office.
No.
Yeah, you freak Robert out. Okay. See you
Great.
guys.
Thank
Have a
you.
nice
Bye. Have a good
weekend.
night. Have a good
Next
day.
weekend.
Good night.
Bye. Good night.
We start playing the roll call stops in before we